This PowerPoint describes the connection between hemoglobin synthesis and the presence of microcytic hypochromatic anemia. Errol Nelson, a 55-year-old male, presents with a recurrent rash and with urine that has a blood-like color. Examination of the patient found a rash on his arms and legs. In some cases, there is blistering. The urine has a reddish-brown color, which when acidified and examined under UV light, fluoresced. A mutation was observed in the gene that encodes for uroporphyrinogen decarboxylase. This mutation leads to an accumulation of uroporphyrinogen, which is converted to uroporphyrin. The uroporphyrin migrates to the skin where it reacts with sunlight to produce reactive oxygen species, such as superoxide. The superoxide causes tissue damage, and in this case, the superoxide causes a rash with blisters. This explains the patient's photosensitivity. The uroporphyrin is excreted in the urine, giving the urine a reddish-brown color. It is the uroporphyrin that fluoresces when exposed to acid and ultraviolet light. The diagnosis of porphyria cutanea tarda is made based on a defective uroporphyrinogen decarboxylase. This porphyria is hepatic in nature and is autosomal dominant. The take-home message is that microcytic hypochromatic anemia is always associated with a decreased ability to make hemoglobin. The decreased ability to make hemoglobin could be either from an iron deficiency, an inability to make heme, or an inability to make either the alpha or the beta subunits of hemoglobin. Adult hemoglobin A contains 2 alpha-like subunits, which is shown in red, and 2 beta subunits, which is shown in blue in this particular figure. About 98% of the hemoglobin content in an adult red blood cell is hemoglobin A. The remainder of the hemoglobin is hemoglobin A2, which is an alpha-2, delta-2 heterotetramer. The delta chains are similar but not identical to the beta chains and are encoded by different genes. What I mean that they are similar is the delta chain and the beta chain have 146 amino acids per subunit. They both bind a heme group, and the sequence identity is very high, much higher than, say, between the beta chain and the alpha chain. The hemoglobin, of course, is what gives blood its characteristic red color. Therefore, if we're not able to make hemoglobin, the intensity of that red color is going to be decreased. And this is why we would say that the erythrocytes are called hypochromatic, that is, their color is reduced. Now we're going to start off by talking about iron, which is needed for the synthesis of heme, which is, of course, needed for the synthesis of hemoglobin. And it turns out that iron is the major regulator of hemoglobin synthesis in developing red blood cells. Iron is absorbed by enterocytes that line the small intestines. The iron atoms are absorbed in its ferrous state. These ions enter the cell through the divalent metal transporter 1, a process that requires co-transport of proton ions. Ferric iron needs to be reduced to its ferrous state, a reaction that is catalyzed by duodenyl cytochrome B. Once the iron is in the enterocyte, it is oxidized and stored in the protein ferritin, or it can exit the enterocyte through the channel ferroportin. Heme groups enter the enterocyte through the heme carrier protein 1, where it can be degraded by the heme oxygenase, either heme oxygenase 1 or heme oxygenase 2, forming biliverdin, carbon monoxide, and iron, which can again be stored in its ferric state within the protein ferritin. Once stored in ferritin, it can be released and reduced again as a single step, where it can exit the cell through ferroportin and then be reoxidized to its ferric state by the protein heptastin. Ferric iron is bound by apotransferrin where it forms transferrin and transported to the tissues that needs the iron. Now, this slide kind of describes the regulation of hepcidin synthesis and secretion. The iron that is carried by transferrin combines to the transferrin receptor 2, signaling a state in which the iron concentration is high. This binding event activates the atypical major histocompatibility class 1, abbreviated HFE, and hemojuvelin, which is abbreviated HJB. These two proteins act together and cause the increase in transcription of the hemp gene, leading to the production and the secretion of hepcidin. Hepcidin travels through the blood to the enterocytes and macrophages, inducing these cell types to transport the ferroportin receptors to the liposomes where the receptors are degraded. Iron that remains within the enterocytes will be lost from the body when the enterocytes are sloughed off. Hemochromatosis occurs from a mutation that prevents functional hepcidin from being made. Inactivating mutations can occur in hepcidin that directly alter the structure and function of this hormone. Mutations can also occur in HFE and HJB. These mutations inhibit the synthesis of hepcidin by preventing a functional transcription complex from forming. More rarely, mutations can occur in transferrin receptor 2, preventing the hepatocytes from sensing serum iron levels. In each case, hepcidin levels are decreased, preventing this hormone from degrading ferroportin, thereby allowing iron to freely enter the body. As doctors, you will more likely encounter patients with an iron deficiency than a patient with hemochromatosis. Therefore, you will need to be able to suggest foods that contain iron. Now, iron is present in a number of meat sources, mainly due to the presence of myoglobin. And another good meat source, is liver, mainly because it contains large quantities of cytochrome P450. However, in our case, we have a patient that's a vegetarian. In this particular case, you will need to suggest options that are vegetarian in nature. So therefore, you need to know the vegetarian sources as well. These sources include spinach, beans, and dried fruit. Of course, there's also many other factors that can impact iron absorption. For example, ascorbic acid will reduce ferric iron to its ferrous state, and this will improve its ability to be imported into the enterocytes through DMT1. In contrast, divalent ions such as calcium will compete with iron for entry into the enterocyte cell, and therefore that will limit iron absorption. It's good. These are just two examples. There's several more that's presented in the book, and you need to remember these examples. Now, this figure provides an overview of iron metabolism from a hematology perspective. Iron enters the body through the enterocytes in a process that's regulated by ferroportin and hepcidin. Iron is transported to the hepatocytes and erythrocyte precursors by transferrin. And then iron is transported into the erythrocyte precursors by the transferrin receptor 1. It is first incorporated into the protein ferroketolase and activates the transcription of allosynthetase 2 because of the presence of an iron-responsive element in the gene structure. The increase in the enzymatic activity of allosynthetase 2 and ferroketolase leads to the synthesis of heme groups, which in turn activates globin synthesis by stimulating the translational factor EIF2. The erythropoiesis increases the presence of growth differentiation factor 15, which is abbreviated GDF15. And high levels of GDF15 will repress hepcidin synthesis in hepatocytes. And high levels of iron will stimulate hepatocytes to make hepcidin. So therefore, the amount of hepcidin made is dependent upon these two signals, and the amount of total iron that's present in the body is related to the concentration of hepcidin in the blood. Now we'll talk about heme synthesis. And it should be noted that heme synthesis occurs both in hepatocytes and the erythroid precursors. I should say it occurs in more tissues, but those are the two major cell types that utilize heme biosynthesis. There are 8 steps involved in the synthesis of heme groups. The starting materials are glycine and succinyl-CoA. The first step takes place in the mitochondrial matrix and involves the synthesis of delta-aminolevonic acid catalyzed by the enzyme delta-aminolevonic acid synthase. This enzyme is abbreviated allosynthase. This enzyme catalyzes the rate-limiting step of heme synthesis, and this enzyme requires pyridoxal phosphate for activity. Remember that pyridoxal phosphate is derived from vitamin B6. In humans, there are 2 allosynthases, allosynthase 1, which is found in the liver, and allosynthase 2, which is found in erythrocytes. A defect in the synthesis of erythrocytic allosynthase 2 causes sideroblastic anemia. This disorder is characterized by microcytic hyperchromatic anemia and iron-laden mitochondria that surround the nucleus. The second step in the synthesis of heme is catalyzed by delta-aminolevonic acid dehydratase, or ALA-D, and this step results in the formation of porphobilinogen. The last step in the synthesis of heme is catalyzed by ferroketolase, which is abbreviated as FC in this figure. This last step results in the insertion of the heme iron into the heme group. Lead, the heavy metal lead, inhibits catalytic activities of both ALA dehydratase and ferroketolase. Lead inhibition leads to hypochromatic microcytic anemia because of the inability to make heme and therefore inability to make hemoglobin. And the inhibition of ALA dehydratase leads to an increase in delta-aminolevonic acid in the blood and in the urine. So these are a good diagnostic tool. And you should note that the buildup of delta-aminolevonic acid in the blood is thought to be neurotoxic because it resembles the neurotransmitter gamma-aminobutyric acid. There's a second important point that's found in this figure. Hydroxymethylbilane, which is shown in blue in this figure, can cyclize in both an enzymatic and non-enzymatic reaction. The product of the cyclization reaction is uroporphyrogen, which can be converted into porphyrogen enzymatically. Both uroporphyrogen and coporphyrogen can be oxidized to their respective porphyrin compounds. When exposed to light, the porphyrin compounds can generate large quantities of reactive oxygen species, such as superoxide and hydrogen peroxide, causing tissue damage and pain. This is one of the main reasons why patients with defects in the enzymes that synthesize hydroxymethylbilane, uroporphyrogen, and coporphyrogen exhibits photosensitivity. So many of the porphyrias that are genetic diseases that have a defect in the heme synthesis can impact either hepatic cells or erythroid cells. They rarely impact both cell types. And the reason for this is because there are different isozymes in each cell line. Now, genetic lesions that impact the erythroid cells will cause microcytic hypochromatic anemia as one of its clinical features. And porphyrias that impact hepatic cells tend to cause abdominal pain. An example of that is acute intermittent porphyria, as shown in this figure. This particular porphyria has a defect in the conversion of porphobilinogen to hydroxymethylbilane, and this leads to intermittent abdominal pain. The intermittent nature of this disease is caused by a variable expression of allosynthase 1. And it's already well known that compounds such as barbiturates and ethanol will increase the expression of allosynthase 1, leading to an increase in the synthesis of heme, which in turn leads to an increase in cytochrome P450. And this occurs in patients without the disease. However, when you have a patient with acute intermittent porphyria, then the presence of barbiturates or ethanol or other drugs will lead to a greater flux through the heme synthesis pathway, but it will get stuck at the lesion, site of lesion, in this case, the inability to convert porphyrobilin into hydroxymethylviolane. This leads to a buildup of porphyrobilin and a buildup of delta aminolevonic acid, or ALA in this particular, abbreviated ALA in this particular figure. Both of these compounds are thought to play a role in the pain, and sort of these compounds themselves are the mediators of pain. And this gives it a biochemical rationalization of why you not only have pain, but you have intermittent abdominal pain. Now I'm going to move on to the synthesis of globin chains. And of course, the inability to make globin chains will be characterized by microcytic hypochromatic anemia. And these particular lesions are referred to as thalassemias. This figure shows the overall pathway of hemoglobin synthesis. And of course, iron triggers heme synthesis, which in turn triggers globin synthesis. The globin mRNA is translated by cytoplasmic ribosome. The beta chains will self-associate into a beta-4 homotetramer, which is known as hemoglobin H. And the alpha chains will first interact with the alpha heme stabilizing protein, which is abbreviated as AHSP. The alpha and beta chains will then associate into heterodimers, which will then also associate into heterotetramers as concentration increases. Mutations that impact the stability of either chain or the presence of either chain will lead to thalassemia. And mutations in AHSP will also lead to an alpha thalassemia type phenotype. This figure gives sort of an overview of the pathology. More often than not, that beta thalassemia is caused by point mutations leading to problems in protein stability, such as hemoglobin E, splicing, and/or transcription/translation. Patients with beta thalassemia will have an increase in hemoglobin A2 because the delta chain is not impacted by the mutation in the beta chain. Patients with beta thalassemia will overproduce alpha chains compared to the beta and delta chains, leading to protein precipitation, membrane damage, and ineffective hematopoiesis. This is why patients with beta thalassemia will require folic acid supplementation. Note that patients with hereditary persistence of hemoglobin F will have milder symptoms because HbF will be present. Patients with alpha and beta thalassemia will have milder symptoms because the alpha chain concentration will be about the same as the beta chain. And a lot of the pathology is because of the increase in alpha chains that are unpaired. Now this figure gives an overview of alpha thalassemia. That is the disorder that occurs when developing red blood cells cannot synthesize enough alpha chains compared to the beta chains that are present. This leads to the accumulation of beta chains that self-associate into beta-4 homotetramers. And note that the beta-4 homotetramer cannot deliver oxygen, and 2 is very unstable and will also precipitate. Gamma chains will also associate into a homotetramer that's called hemoglobin BART during the fetal stage of development. In contrast to beta thalassemia, the disease, the alpha thalassemia is caused by deletions in the genes that encode the alpha chains. And note that there are 4 alpha genes in the human genome, 2 from each parent. The severity of the symptoms is dependent on the number of alpha gene deletions. And of course, like patients with beta thalassemia, patients with alpha thalassemia will develop folic acid deficiencies that require supplementation. And for this phase, you will need to know the food sources for folic acid. Obviously, leafy vegetable sources are a good source for folic acid, and of course there are several other sources, including beans and fruit sources. Of course, the fruit sources being quite a bit, not quite nearly as good as the leafy vegetables and the beans. Now, as far as targets, you need to remember that patients who have that require an increased amount of red blood cell synthesis will need folic acid supplementation. And folic acid supplementation is probably a good idea for people who have the reduced ability to absorb vitamins. A good example of this would be alcoholics and the elderly. To conclude, Harold Nelson presents with a recurrent rash and with urine that has a reddish color. The examination of the patient revealed the rash in areas exposed to the sun. Analysis of the urine found a reddish color that exhibited fluorescence when acidified and examined under ultraviolet light. The photosensitivity and the red-colored urine are attributed to the presence of one or more intermediates of heme synthesis. The genetic analysis confirmed the diagnosis of porphyria cutanea tarda when a mutation is found in the gene that encodes uroporphyrinogen